regions. And only the variable regions are necessary for specific recognition of a foreign protein. And therefore, in the initial model, the assumption was made that this 12 nanometer tall structure was necessary. In fact, it's not. Since the model was initially published, people have learned how to make single chain FVs, and these things are about 3 to 4 nanometers in height. Here we have the variable heavy, the variable light, and here we have the CDRs that contact the antigens on the surface of the single chain FV. And so, in fact, you can substantially reduce the distance between the bound analyte and the sensing surface simply by moving away from these full-length antibody fragments down to an engineered single-chain FV. And so the way that we get the single-chain FV, many of you no doubt are very familiar with it, is that we use a high-throughput display method. And so basically this is a diagram of filamentous bacteriophage in which you have your favorite protein presented as a fusion to one of the phage proteins and encoded by the DNA encapsulated in the virus. And this allows you a very convenient means to select out of a library of variants those proteins which recognize the protein of interest. And so basically the procedure is that you construct a library that has 10 to the 9 to 10 to the 10 individuals, and you take this pooled library, you expose it to your analyte of interest, in our case to MIG, you incubate it, you allow it to bind, you isolate the MIG-bound phage particles, wash away the rest, and then you elute the phage and repeat the process. And what you end up with is that the phage enriched. A larger and larger fraction of the population as the selection goes on is capable of recognizing MIG. So this is just standard phage display technology. Okay? And so for the purposes of engineering this protein for its use in the sensor, there are a couple of things that we want to do. So we wish to be able to use chemoselective conjugation. We want to be able to orient the protein on the sensor surface. And so what we use is an oxyamine conjugation chemistry. All chemoselective conjugation chemistries consist of a nucleophile electrophile pair that are mutually exclusively reactive for one another. And so what one can do is if you have two chemical constituents that contain such a nucleophile electrophile pair, and typically the electrophile is an aldehyde or it's some sort of an alkane group. And then if you have a nucleophile, in this case an amino oxy group, you will form a specific oxime bond only at this site. And one of the things that we discovered early on when we were playing with these proteins is that if you put an N-terminal serine at the beginning of a polypeptide sequence, and then you expose that polypeptide to mild oxidation, the serine is converted to an aldehyde, which provides you now with an electrophile. And so if we derivatize the surface with the SAM that contains a nucleophile, it now becomes possible to do oriented deposition of your antibody fragments. And so basically what we're showing here is the molecular biology just simply to reduce or to replace the N-terminal alanine of one of the single chain antibody fragments with the serine, and then subsequently oxidation and expression, and you are able to then generate a protein which will participate in the chemoselective conjugation. And this is mass spec data that shows that in fact the chemoselective conjugation is very specific. So for the individual proteins, what we show here is with the sear-terminated proteins, there is a single residue that undergoes a predicted size mass shift when one does a light oxidation of the protein. So here we have the unoxidized form, and here we have the oxidized form. And basically, we get a mass shift of the expected molecular weight. It's about 16 atomic mass units. And that indicates that when we oxidize the protein, we are only changing a single residue. And you can delimit where that residue is by trypsin digesting the polypeptide and then running the mass spec again and asking in which fragment the mass shift occurs, and it occurs in the N-terminal fragment. So in fact, what you're doing is producing a aldehyde that occurs only at the N-terminus of the polypeptide, and what that means now is that I'm capable of doing chemoselective conjugation of this single-chain FV onto the surface. And so there are a couple of things that you could do that would reduce the critical distance between bound analyte and sensing surface to enhance the sensing signal. And a couple of them are very simple. One is that when you do these phage display methods, what you always get is a selection of different ligands that recognize whatever your target protein is. And so, in other words, in the case of the single-chain FVs, these are things that recognize different MIG epitopes. And so, in principle, the consequence of having receptors that recognize different epitopes is shown here. So if you hypothetically say on MIG that you've got 2 epitopes, which is untrue, there are probably 8 or 9 groups of epitopes in a protein this size that are not overlapping. But basically, this shows an assay that would allow you to determine that, in fact, you have more than one antibody specificity for epitope. But the interesting thing about it is, obviously, the charges on MIGs are fixed in space, and the epitope positions are fixed in space. So if I use as a receptor an epitope or a antibody that recognizes a particular epitope, and compare that to another antibody that recognizes a different MIG epitope, one would expect that the charges on the surface of MIG would be moved relative to the sensing surface. So this is one way in which one can tune the sensitivity of the device, but you can only take advantage of this if you have access to chemoselective conjugation. Okay, another way in which you can take advantage of, I'm going to fill this in a little bit. This is a model of MIG, and what this shows is the white residues, it's a space-filling model, are uncharged amino acid. So the blue residues are positively charged, the red residues are negatively charged. And so if you allow, if this is my single chain FV, and it's down on a surface, if you allow this to be the single chain FV, depending upon which epitope my antibody recognizes, I would expect that it would bind MIG either in this sort of configuration or possibly in this sort of configuration or possibly in this sort of configuration. But what you can see is that I'm moving around the position of the charges relative to, this sensing surface, right? So I can adjust proximity this way. Another way in which one can adjust proximity, which surprised the living daylights out of it when we first did it, is that polypeptides in particular are extremely amenable to circular permutation. So what is circular permutation? Well, think of the linear amino acid sequence. Imagine that you join that into a circle, right? So you seal the N and C ends, and then you create another N and C end somewhere else on the surface. So you're moving around the domains of the protein relative to each other. And what we found when we did this as a phage display exercise, we initially did it with a 4 helical bundle protein, and it was GCSF. But what we found was, remarkably, out of the library of variants, about a half of the circularly permuted forms of the GCSF molecule were fully capable of engaging receptor. So they were functional, and this was a surprise. This is a rather gross perturbation of the protein sequence, and yet we retain function. You can do the same thing with antibodies, and the consequences are shown here. And so here we have our single chain FV. This is the variable heavy variable light. These are the CDRs in pink. And what you can see is I've used chemoselective conjugation that hits only at the end of the polypeptide. And just as is the case with the circularly permuted 4 helical bundle protein,